African Center of Excellence for Genomics of Infectious Diseases, ACEGID, is a World Bank-funded center of excellence established in 2014 to bridge the shortage of skilled personnel and adequate tools for research and innovative solutions to Africa's challenges. The ACEGID platform was set up to train the next generation of African genomic scientists to help understand circulating health threats and facilitate effective, evidence-based outbreak preparedness and response.
The vision behind establishing and founding ACEGID is a vision that I've had over, I will say, 25 years. And the goal was to set up a platform where devoid of outside influence, where that is a platform that is driven by academic and research, where young Africans could come and conduct relevant, highly translational and ethical research for genomics, for public health and infectious disease in Africa. So that was the goal. And then that platform is supposed to be an open platform that transcends national boundaries. And the reason why we set up this international platform is simply because we also realize that in Africa, knowledge is caged. There's no brain circulation. So we really wanted a platform where young Africans people could come from everywhere in Africa and then come and work and collaborate and create, this ecosystem where people can, talk to themselves and use genomic knowledge to address, African problem. And infectious disease actually being one of those major problems. Specifically, we wanted to use such a platform to to build a critical mass of well-trained young African scientists that will use the skills and the knowledge of genomics to address, infectious disease problem on the continent.
ACEGID is a consortium of West African academic and medical institutions with long-standing partnerships in research and training. ACEGID's mission is focused on education and research. The education and research missions of ACEGID fuse into genomics capacity building, that is, raising a critical mass of well-trained African scientists who are capable of conducting independent research on infectious diseases.
It's not all about the space. It starts from your mind. It starts from what you're thinking. We have a small space, not a very large space, but technology has evolved in a way that you have most of this equipment come in like very cute small sizes. And so we've been able to maximize the small space that we have. And, it's about what the equipment can do, not how bulky it is.
In 2014, ACEGID confirmed the first case of Ebola in Nigeria within 6 hours, a singular feat that led to the early and successful containment, management, and control of the disease in Nigeria. ACEGID released the first SARS-CoV-2 sequence in Africa within 72 hours of receiving the sample, having completed the sequencing in 48 hours. The World Health Organization and Africa Centers for Disease Control and Prevention jointly designated ACEGID as a reference laboratory for the genomic sequencing of SARS-CoV-2 and other emerging pathogens. Within 1 year, the center trained more than 100 scientists from over 30 African countries in SARS-CoV-2 sequencing and bioinformatics. Scientists trained by ACEGID lead the sequencing efforts of their respective countries. The center's postgraduate programs are equivalent to those in European countries, and evidence of this is the accreditation by the Agency for Quality Assurance through Accreditation of Study Programs, AQAS.
So my background as a veterinarian, there are certain parts of the veterinary practice that has to do with diagnostics. And other diagnostics, there are certain aspects where you talk about the molecular aspects of diagnostics. And I remember then as a 400 level paramedical student, and I wasn't really sure what this aspect holds or what was the promise in this particular area. But coming down to ACEGID, it has really helped me to wrap my head around the promise that this aspect actually has to offer science in general and even innovation. I saw that sequencing was being done. You don't have to send out your samples anywhere for anybody to sequence them for you. Then going beyond that, I found out that even analysis of the data could be done right here.
Working with our partners, we developed rapid testing kits that determine the presence of Ebola virus and Lassa fever virus within 10 minutes. The center continues to drive innovation by using genomics for surveillance, characterization, and diagnosis of infectious diseases, as well as for vaccine and drug development.
SHINE is a CRISPR-based diagnostic technique, which was designed by our collaborators at the Sabetila Broad Institute. So the whole idea of SHINE is to be able to detect SARS-CoV-2, to be able to have a point-of-care diagnostic tool for detection of SARS-CoV-2. We are going through a lot of validations for SHINE so that it can be as rapid as your malaria test kit. But the difference between SHINE and your malaria test kit is that it looks out for the nucleic acid for the SARS-CoV-2. And this technique or this technology can also be employed for other RNA viruses.
We work actively with national, regional, and global partners as we use genomics tools and techniques to push the frontiers of infectious disease research on the continent.
Our partners include our funders. Our partners also include the people that we work with, the institutions, other research institutions, other well-known research organizations across the world that we work with, that will make us to bring, that cause us to have the kind of impact that we have in research. The clinical sites where we work here in Nigeria include the General Hospital Ikorodu. We're working with Iwa Specialist Teaching Hospital in Edo State. We're also working with Federal Government Hospital, Federal Medical Center, Owo. We're also working with Alex Ekome Federal University Teaching Hospital, Abakaliki.
We have a team of veterinarians that are working, in the bushes. They are actually sampling animals and then sampling animals that are living in close proximity with humans. And then we basically just using genomics as a way to really scan through the blood samples of those animals to see exactly what pathogens are present and if those pathogens are infectious and then how they spill over from those animals to human. We're already in the bushes. And we really use this very innovative One Health genomic approach, to study, disease spillovers, and then also to really see how we can, learn from those studies to prevent, future epidemics and pandemics from happening.
ACEGID ensures mutually beneficial engagements with our partner sites. The center has established modern genomic sequencing hubs and diagnostic sites in different parts of West Africa, including Nigeria, Senegal, Sierra Leone, and Liberia. These partners now have the capacity to identify and analyze unknown circulating viruses, and they constitute the most advanced viral sequencing capacity in the sub-region. Working with partners at the Broad Institute, ACEGID is implementing Sentinel, a pandemic preemption and response system that aims to stop pandemics before they start.
We have this big program called Sentinel, which is really an early warning, system for pandemic response, for pandemic preemption and response. And that is really built on on three major pillars. Those pillars are early detection of pathogen that we know and then we don't know. Connection of this information in real time to public health authorities. And then the third pillar, which is empower, which is really building the strong workforce and equipping the workforce and building the capacity of the workforce to use genomic, diagnostics, the next generation of genomic diagnostics that we're developing in order to detect pathogen early, but also to provide that information in real time to public health authorities. And then once we have those two things, we can eventually overcome and contain epidemic as they start.
Africa is home to 7 out of 10 pathogens with pandemic potential. At ACEGID, we consider these pathogens blessings in disguise.
Africa is blessed with not only natural resources, not only human resources. Africa is blessed with a very powerful biodiversity. And as part of the African biodiversity are, pathogens. They are part of our biodiversity. And as such, they shouldn't be seen, these pathogens shouldn't be seen as just challenges or drivers of disease. They should also be seen as blessings in disguise. Why am I saying blessings in disguise? Because yes, God wasn't stupid to actually bless us with those pathogens, because it's actually paved the way. Those pathogens provide opportunities, and these opportunities should be that we Africans should be in the driver's seat in terms of finding solutions, to infectious diseases. Because with the pathogens are part of our own environment, part of our ecosystem. So we should be the ones, studying those pathogens, providing answers and finding countermeasures. And those countermeasures could be diagnostics, could be therapeutics, could be vaccines. And that's the reason why we should be hunting pathogens. That is, understanding those pathogens and characterize them, developing countermeasures before they come to us. So that's really the whole idea about creating this platform, or what we call the next generation of African pathogen hunters, because we have to do that, and then we need to do that in the dirt roads of Africa, in the bushes of Africa, and we need to really equip them so that they should be able to, characterize, know those pathogens, characterize them better, those that we know, those that we don't know, before they come to us. And then that's really, we should be the frontliners, and not only for Africa, but for the world.
We are committed to harnessing the benefits of these pathogens. We are proud to lead the charge to play offense against these pathogens, helping Africa and all humanity to stay ahead of them. You're welcome to partner with us.